Rahim 9700. This is a paper 5 which we're going to solve, and this is the June 19 paper 5 too, and hopefully we should finish both the questions in this video. If not, then we'll go on to the next video. As I've said in previous paper 5 videos, this is for 1 hour 15 minutes and it's only for 30 marks, while on the same day you have paper 2 which is also for 60 marks. So this is 30 marks in the same time, and the reason being is that you need to think and you need to really read the questions carefully and understand them before you start answering them. So please take your time and answer befittingly as you read the question and understand the question. Some students wanted to determine the reaction time of a sample of people aged from 5 to 80 years. So that's a very, very large range. 5-year-old and 80-year-old makes a lot of difference. The students found three different methods of testing reaction time as described in figure 1.1. So they found out that there were three different methods by which they could test the reaction time. Now if you look at method 1, it's a ruler test, and we've seen this in O levels as well. The subject rests their elbow on a table so that their wrist extends over the side. The assessor holds the ruler vertically in the air between the thumb and first finger of the subject. The zero mark on the ruler is lined up with the first finger of the subject. The ruler is released without warning. As soon as the subject sees the ruler fall, they catch it as quickly as possible. The distance the ruler falls is recorded in meters. The distance the ruler falls is recorded in meters. Then method 2, computer click timer. The subject looks at a computer screen, looks, eyesight, and clicks a start button on the screen and waits for the background color to change. As soon as the background color changes, the subject clicks a stop button on the screen. The reaction time on seconds appears on the screen. So here the reaction time is measured in seconds. Here the distance falls and recorded in meters. So look at what we are measuring in each test. Then method 3 is a light board test. The subject stands in front of a board with buttons arranged in a pattern. The buttons light up randomly. As soon as a button lights up, the subject presses the button to turn it off. As soon as a button is pressed, another button lights up. The number of light turns off in a set time is recorded. The number of lights turned off in a set time, so set time may be 1 minute or 30 seconds or 1.5 minutes, is recorded. So you need to do this question, you need to understand this on your own and read all of these carefully. And just like I'm mentioning this, you have to imagine that you're actually doing these things and how would you manage this. Now, when we look at the first part of the question, it says the student decided that method 2 was the most suitable to use. The student decided that method 2 was the most suitable to use for testing subjects aged from 5 to 8 years. Suggest two reasons why method 2 is the most suitable. So when you have to say when it's not suitable or suitable, I've told you this before also, is that you have to find faults in it. Here you have to find the good things about it. And in the good things, the computer click timer, the subject looks at a screen, clicks a start button on the screen, and then waits for the background color to change. As soon as the background color changes, the subject clicks a stop button on the screen. And the reaction time in seconds appear on the screen. In seconds appear on the screen. So everything is very automated, everything is very there's not much that the human intervention has to be done. Somebody holding the ruler and then letting the ruler fall and then somebody catching it. Here we have a board. So you have to think of the goods and bads of each of them and then come up with, okay, what's good about the method 2. Now, part 1 of the question, it says the student decided that method 2 was the most suitable and then suggests two reasons: easy to explain, easy to do. Physical strength or stamina is not necessary. We didn't have to catch a ruler and we didn't have to really, have a lot of issues there. No calculations needed to get reaction time. And then results are more precise. Well, I mean, the computer was generating all this. So all these were the plus points of the method 2, and you had to really look for them and then word them and put it in the correct manner. This was for 2 marks, and you would have to write something very sensible and word it very correctly. Then state the independent variable. I've always told you independent. I changed this. So what did you change? You changed the age of the subjects. They were anything from 5 to 80 years. So this is what you changed. So that is the independent variable. And what did you measure? You measured the reaction time. That is always the dependent variable is the result and what you're going to measure in the experiment. Whether you're going to measure the bubbles of oxygen gas produced, or are you going to measure the volume of gas produced. So whatever you're going to measure, you're going to measure the reaction time. In my first video on paper 5, I have mentioned what is independent and dependent variable, so please do go over that as well. Then part 3 of the question is your designing and planning. Describe how the students could investigate the reaction time of a sample of people aged from 5 to 80 years using method 2. Your description should be set out in a logical order and be detailed enough to let another person follow it. Details of method 2 as described on page 2 are not required. So you do not need to repeat the words of the question and give me again the method 2 which was given in the on the previous page. So now you have to think about how, and as I tell you always is paper 5 is how will you be doing this in the lab. So you have to visualize and you have to just think if you had to do this experiment, how would you do it? Now, if you look at the details of method 2, as I told you, we would do this. So let's first select the number of people we want to take. So we were going to select people between the age of 5 and 80. So suitable number of people in total, maybe you want 100 or you want 80 of them. So equally spread age categories. You can't have all of them 10-year-olds or all 50-year-olds or all 60 years old. So I would say you divide it into 3 or 4 categories and then you have 20 in each age group. So I've just given you a rough example of that. Age 5 to 30, or you could even do less than that. You could do 5 to 15, and you can make 5 groups and take 20 in each group so that you have a spread of the data, so that you have uh, data from each age group. Because you see, they asked you to do something between age 5 and 80. So between 5 and 80, I've made 3 groups, but you could, of course, come up with more than those. Then same number of males and females in each group. You see, you can't be having only 5 to 30 only men and then 31 to 55 only females. Well, you couldn't be doing that. So same number of males and females in each group. So if you were going to decide this, you would actually be thinking about all this. And then you had to have a consent form because you were having 5-year-olds as well. So you would have to have a consent form that you were going to use this data and you could do this experiment with the children, or you could do it with the older people. And the older people were also informed that this experiment was going to be run and then this data was going to be used for some research. Then same type of the computer or the tablet or the software or the same color change. So all that should have been standardized and that would have been 1 mark. That won't have been 2 marks. So same type of computer or a tablet or the software or the color change. Then the same room conditions. If we were going to do it in the computer lab, or were we going to do it in a big hall, or how were we going to do it? Where were we going to do this experiment? So same room conditions, the same temperature, the furniture, computer lab, or any other place which you would think of. And then, of course, you should be avoiding people with a neurological deficit or a poor eyesight, or maybe if they're colorblind. So you would not do this experiment on people and embarrass them by selecting them. And then, when they made a mistake, and then you would say, oh my God, this doesn't go with the readings, but they were actually colorblind and they could not see the colors. So you would get a very weird result because the data would be skewed. You'd have some data coming this way and then some, because the people were colorblind were going to do it, were going to mess it up. So then we would ensure that there were no people on drugs. There could be people taking some neurological issues, and they could be taking some medicines, or they could be on caffeine, or they could be alcoholics. So all those conditions would be ruled out when we selected the people for this experiment. Then we would test at the same time of the day. Testing at the same time of the day or a stated time like 12 noon, or maybe 10 in the morning, or maybe 3 in the afternoon. So you would have to state a time and say, okay, well, this is the time that we would do the experiment and we would do it in this 1 week, because you couldn't do all of them at one time. Maybe you could. Then using the same hand to push the button. So this is all, when if you think if you're actually going to do it, then you would think of all these points. then minimum of 3 tests on each subject and taking a mean. Or you could say minimum of 3 subjects in same age category and taking a mean. So you could have come up with that as well. So this is what we had to do, and then this was a low risk experiment. Why? Because the parent or guardian present for the children, or if there were people who were older, then a carer for the very, very old people who needed some sort of help and or they were on wheelchairs or something like that. So you needed a carer for that. So these were a number of, they were about 12 mark scheme points, and you had to only give me any 6 out of these to get your 6 out of 6. So unless you all start thinking on these lines and think that you're actually going to do this in the lab, only then you could have come up with these 6 points, and I'm sure you all could have done that, and you could have got your 6 out of 6. Then we come to the part B of the question. The students found some results on the internet for a reaction time test as shown in figure 1.2. The error bars show plus minus 1 standard deviation. And mean reaction times is on the y-axis, and we have the age in years on the x-axis, and you have a separate one for male and a separate for female. And the percentage change in mean reaction time for males between the ages of 5 years and 10 years is 47%. So this is the information they're giving you. And calculate the percentage change in mean reaction time for females between the ages of 5 and 10 years. Show your working and give your answer to the nearest whole number. Now we don't read that many times, and that costs us. And this is 2 marks, and nobody should lose these 2 marks. They're very, very important 2 marks, and everybody must get these 2 marks. And it's very easy to do this calculation because it said calculate the percentage change in mean reaction time for female. So you have to circle the female. This is what I've just done. And between the ages of 5 years, so this is 5 years, so this is here we are 5 years, and then it said 10 years, so 5 years and 10 years. So here we've got the 10 years. So in between this, now here you can see the female is 300, this is 300, so this is 350, and then of course you've got to figure this out, so this would be 340, and then this was 210. So of course it was easy to do the calculation, but I mean I'm sure we would all be having problems in doing this. So 340 minus 210 over 340, and of course you multiply it by 100 to get the percentage change, and this is what you get. It is 38%. So 340 minus 210 over 340, so it is the change over the original into 100, and this is how we calculate a percentage change, which everybody must know how and why do we do this. Now in this part 2 of the question, what are they asking you? They're asking you state 2 conclusions about the trends shown by the results in figure 1.2. Can we look at figure 1.2 again and then just assess the trends? Closely at figure 1.2, and you see something very remarkable which everybody should be able to look at when you're looking at a graph. When you're looking at a graph, you must be looking at the trend. Now, if you look at the trend, it says mean reaction time in females is higher than in males at most ages. Here only we see a little change here, but otherwise, what do we see? We see mean reaction time in females is higher than in males at most ages. You see, we're only seeing a slight deviation here at this point. But I mean, let's not look at it. But generally, what do we say is mean reaction times in females is higher. I've colored the female red and I've left the other one black. Then the second point was reaction time decreases to age 20 years and then starts to increase. So you see here till 20 years, it starts to decrease and then it starts to increase. So mean reaction time decreases to age 20 years and then starts to increase. Then due to the overlap of the standard deviation lines, you see these are the standard deviation lines which you can see here. There is an overlap, and then there is the other one which is also being shown here. So I mean, you see a standard deviation, there is overlap. So due to the overlap of the standard deviation lines, there is no significant difference between male and female reaction times. You see, when the standard deviation overlaps, then that means that there is no significant difference. So these are the three mark scheme points which you were supposed to give for this, which shows the trend of it. I have worded it so mean reaction time is in sorry, now the first point was mean reaction time in females is higher than in males, and I've just shown it to you on the graph. Then the second point was reaction time decreases to age 20 years, then starts to increase. And the third point was due to overlap of the standard deviation lines, there is no significant difference between male and female reaction times. Then as you look at part 3 of the question, state a suitable statistical test. Now, whenever we are comparing two sets of means, we always do the t-test. So this is what we had to say is the t-test to find out the difference in reaction times between male and females of the same age was significant. So you do the t-test. Why do you do the t-test? Because you're actually comparing means, or you could have said data is continuous, or you could have said standard deviations are approximately the same. So that is the reason why we would choose to do the t-test. Then the next and the last point is suggest a null hypothesis for the test that you have chosen in part 3. Now, there is no significant difference. Null hypothesis means you always negate it. So, there is no significant difference between the mean reaction times between males and females at the same age. So, because you see, we had to take it from here that we were talking about the males and females at the same age was significant. So, the null hypothesis would be to negate it and say that there is no, and this is why I've underlined the no part, there is no significant difference in the mean reaction time between males and females at the same age. And the time was underlined, and that's why I have underlined it, which means the word time had to be used. So that finishes this first question. Now we start the second question. Now, as you look at question 2, it was a more complex question. And sperm production in humans is controlled by the hormone FSH and LH. Of course, these are female hormones also. We talk about them in the menstrual cycle, but here they're talking about in humans is controlled, and these hormones are released from the pituitary gland in males to maintain a constant concentration in the blood. And we're talking of sperm production, so we're talking of what is happening inside the testes. Research has shown that sperm production in humans has decreased during the last 75 years. The research has highlighted concern about the fertility of future generations. Number A part asks you, assessment of fertilities are carried out on diluted semen samples. The samples are checked for number of sperms per millimeter cube, motility, are they motile or are they not motile, and any abnormal structure. Do they have the flagellum or the tail? Do they have some have head, some have not half the head, or some have something missing? So abnormal structure. Figure 2.1 shows sperms in one part of a hemocytometer grid used for counting sperm. The microscopic illumination causes the sperm heads to fluoresce. Now we must know what is a hemocytometer, and at the end of the video I will show you a diagram of the hemocytometer, and you can then have an idea what we are talking about. It's got a depression in the center of a slide, and there's got a grid at the bottom of that depression. So we can see this, and you can see you should have seen a hemocytometer, or you must have seen it or talked about it. Now it says sperms, and then it says one complete counting square is 0.05 into 0.05 into 0.10. Now the semen sample shown in figure 2.1 was diluted by a factor of 100. Now please understand that diluted by a factor of 100. Describe a method by which the number of sperm present in this diluted sample could be estimated. So count the sperm inside the square. Include the sperm with the heads inside the square, and include those touching the line on the top. So this is what I have done. This is the square, and this is the top line, and this is the left line. So all those touching the line on the top and the left, because the next square, the top and the left would be different. So in this square, adjacent to it, then this would be the top and this would be the left. So you're not going to double it. You will just keep on doing it for each square so that you don't count the sperms twice, once in one square and then again in the other square. So include those touching the line on the top and the left. Then calculate the volume of one square. Now, volume of one square was easy to calculate because all you had to do was you had to just figure this bit out. 0.05 and all this. So you had to just multiply each of that and you got your, so 0.05 and 0.05 and 10, or you got this and you got this value. So calculate the volume of one square, and if you know the number in that one square, so you could then find out and the actual, because you know you diluted it as well. Then part 2 of the question says describe how you could estimate the number of sperm per millimeter cube in the original undiluted semen sample. So volume of one square, divide by the number of sperms in n squares and by volume in n squares, and then multiply by the dilution factor, which is 100. Then we come to the third part of the question, suggest why this method of counting sperm may not represent the actual number of sperm in the original undiluted semen. Yeah, because you only used a very small volume was taken, and the sperms were moving around, so they're very difficult to count. Or the sperms could have maybe formed clumps and they would overlap, and then there was more at one area and less in the other area. And the fourth point was that this was not repeated. We did not repeat the test. So we only did it once. So this is the 4 mark scheme points, and any two of these was going to get you your 2 out of 2. Then the B part of the question now is asking you hormonal imbalances can result in reduced sperm production. So hormonal imbalances reduce sperm production and immobile sperms, leading to infertility. A study was carried out on 120 men. Now that's a very small number with different causes of infertility. All the subjects were non-smokers, did not drink alcohol, did not take any medications for 6 months before the study. The concentration of the hormone FSH and LH were measured in each man. Semen samples were collected and tested within 2 hours of the collection. The number of sperm per millimeter cube were estimated and the motility of the sperm assessed. I did identify two variables that have been standardized in this study. Yes, all were non-smokers. No, none of them was an alcoholic. They were not taking any medication, and the sperm was tested. Within 2 hours of the collection, and the sperm was counted in a fixed volume. So these were the things which were standardized, which were given to you in the question, and you have to just pick those from the question, that these were the facts that they were standardized. So two variables that have been standardized in this study. So you just have to pick things from the question. Then figure 2.2 shows the concentration of FSH and LH in 4 test groups. In one test group, the men are fertile, and the other 3 groups, the men show different types of infertility. So the first one was fertile. This is the first one that we have here. Then the second was infertile due to non-motile sperms, and this was here. And then we had infertile due to low sperm number in semen, and this was the third group. And the fourth group was infertile due to no sperm present in the semen. So this was the fourth group, and this is the fourth group here. So I've just given it different colors to make you understand, and this is what you need to do when you're reading the question. Now state the reason for including the hormone concentrations of fertile men in figure 2.2. Naturally, we would need to compare it with somebody. So we'll have to compare the infertile, because these three were infertile. So we had to compare it with what was the normal and what was the abnormal. So in order to do that, we would have had to have some sort of a set of data from the fertile. So that is the first part of the question. State the reason for, so for a comparison. Now let's look at figure 2.2. Now what do we see in 2? Here we see that the FSH is higher. So let me give it a color here. So FSH is higher here, FSH is lower here. Here again we have FSH about nearly the same, and then of course here we have FSH very, very high. So let's look at FSH in all the 4, and then let's look at LH in all the, so this was the normal. Then this is slightly lesser, and this is now nearly the same, and this is of course very, very high. So if we do a comparison, you can see the comparison here was very glaring, and if you just looked at the graph, higher FSH and lower LH causes group 2 infertility. Higher FSH and lower LH causes group 2 infertility. Then high FSH and normal LH causes group 3 infertility. And then FSH and LH both high in group 4 infertility. So basically just looking at the graph, you could give all these conclusions which you were going to make of the FSH and LH story. Let's look at the last part of the question was C. Semen samples may also be tested for viability of sperm. Viability is the proportion of live sperms present in semen samples. So viability of sperm can be tested using a chemical which stains living cells a different color to dead cells. So the living ones would be different color and the dead ones would be a different color. So out of the total, we would know how many out of 100, how many are living, 20 are living, so 20 over 100. Suggest a method using the stain and a hemocytometer that could be used to test the viability. So that was easy. Record the number of live and dead sperms, or you count the number of sperms of the two different colors, live and the total, and then you of course calculate a method to you give me a method to calculate the proportion. Now, this is what is a hemocytometer. And what we need to understand is a hemocytometer is the name given to a counting chamber designed to count the number of blood cells in a blood sample. Of course, we were using it for that purpose in this experiment. And hemocytometer has a central platform with grooves on either side. There's also a grid attached which resembles a graph paper. And the total volume of the hemocyte is exactly 0.004 millimeter cube. So look at it, there's a cover glass and you can see the side view, there's a mounting support, there's a counting chamber, there's a filling notch, and there's an overflow area. And then of course this is the grid and we exactly know the volume which is going to fit into it. So this is a hemocytometer and we should all be very familiar with it. And that finishes this question on paper 5. Hopefully I should be uploading a few more papers in paper 5 and wishing you all the best of luck in your exams and may you all get the best grade that you can achieve.